let's get started for today. I'm Doug Christensen. I have a joint appointment in electrical engineering and computer engineering and in the biomedical engineering department. And it's my pleasure to introduce a co-worker of mine, Dr. Taylor Webb from the Radiology department. A few things about Taylor's background. Taylor got his bachelor's degree and his master's degree in electrical engineering at it's a little hard for me to read.
BYU.
Oh, BYU.
That's why. I was hoping you wouldn't say that.
It's a little hard for me to read there. He did his Ph.D. at Stanford University, where he worked with passing the ultrasound beam through the intact skull, called transcranial ultrasound, and he'll talk about that. After his Ph.D., he did his postdoctoral training here in transcranial ultrasound for neuromodulation, which is the topic of his talk today. Taylor and I work in the same research group over in radiology, working on the use of ultrasound, not only to image things, but more likely, for this particular case, to do neuromodulation or to do ablation of tissue. It's a very active research group, so it's my pleasure to introduce Dr. Taylor Webb to you.
Thank you. Glad to be here. I apologize for my black eye. I went over the handlebars of a bike on Thursday. It was very exciting, pretending like I know how to mountain bike better than I actually do. But glad to be here. So like I said, I'm going to talk about transcranial ultrasound, focus on neuromodulation, why I'm really excited about ultrasound's ability to give us these kind of all this at once. Targeted, non-invasive, focal therapy in the deep brain. So why am I excited about this? Just a couple obligatory background slides, but also, I mean, this really is why I'm excited about it. It depends how you measure it, but somewhere between like 1 in 3 and 1 in 5 Americans will be diagnosed with a mental illness in their lifetime. 1 in 4 is impacted by a neurological disorder at some point. It could be tremor, it could be epilepsy. Huge burden economically. More important, it's a huge burden of human suffering. And I care about mental illness in particular because I get very excited about helping people get back in the driver's seat of their own life, right? I mean, some of these mental illnesses can really impact someone's ability to chart their own course, and I get really excited about giving them some of that capacity back. So lots of good reasons to care about this. We have ways to do it. The most common way to treat a lot of diseases is pharmacologically. You give somebody a pill. And this works pretty well, actually, for a lot of patients, right? We don't want to undermine that we've made great strides in treating both mental illness and neurological disorders with pharmacological drugs. One of the problems, of course, is you take a pill, it affects the whole body. That's what's represented by the whole body being red here. So if we want to think in terms of precision medicine, we'd rather do this. We'd rather affect the particular part of the brain that's impacted, right? So if we have a mental health disorder, and we know that a particular deep brain network is involved in mood, what if we could just target that network, right, instead of the whole body? Then we might be able to get rid of a lot of the side effects that make these drugs onerous for patients. So that's the promise of transcranial ultrasound. So transcranial ultrasound uses an array of transducers. I've represented that here with this dotted line. Each dot in the line represents, you could think of a transducer. Transducer just converts electrical energy to acoustic energy, right? And so now we can send a wave in, and we can take advantage of the superposition of waves, and we can get a focal effect. So if all the elements are in phase at a particular spot in the brain, we get a jump in the pressure right there, and everywhere else is unaffected. And so this allows us to get 3 things at once, and this is what's really unique about ultrasound. We can get to a deep location, we can affect it in a focal manner, and we can do it non-invasively. As far as I know, ultrasound is really the only technology right now that can do all 3 of these things simultaneously. There are other technologies that can do 2 of the 3. So here's an example. This is deep brain stimulation. This is where we actually put an electrode in the brain. Okay, this is obviously deep. We're affecting it. This is a CT scan of a patient with 2 of these electrodes in. We're affecting a deep location, right? It's focal because it's just where the current density is, right around the electrode. But it's obviously not non-invasive, right? The picture makes that pretty clear, right? We've drilled a couple holes in the skull. We've gone through all the brain tissue from where we drilled the hole all the way to here has been affected, right, as we drop that electrode in. And so we've got depth and focal, but it's not non-invasive. And one of the consequences of this is it's inflexible, right? So you take a disease like depression that's really has a lot of different manifestations, right? It may be that different patients respond differently to targeting different nodes within the networks that affect mood, right? One patient might respond better if we target the anterior cingulate cortex. Another patient might prefer a different target, right? We're not going to try multiple targets with this. We're not going to drop an electrode in here. Oh, that didn't work. Let's just drop another electrode in over there, right? And of course, it's costly and has lots of potential side effects. There are a couple other approaches that are non-invasive. So we've got electrical approaches. We can put electrodes on the scalp and drive currents, and that affects the brain. Or we've got magnetic. We can put a magnetic coil near the head, and we can drive currents through that and have alternating magnetic fields, which of course create currents in the brain. These are non-invasive approaches, but they can't get us focal activity at depth because we get the majority of the action happens near the applicator. Basically, these are fields we don't get a nice superposition effect. So our strongest effect is going to be near the device, which means we can maybe get some focal effects in the cortex, the surface of the brain, but we're not going to get focal effects deep in the brain with either of these two approaches. So again, that's why I'm so excited about ultrasound, because we can do all three of these things at once. And therefore, it's a promising way to try and achieve this goal of precision medicine. So that's exciting, that's great. We can focus ultrasound deep down to the brain. Who says that should do anything? Have any of you been asking yourselves, what does it matter if we focus ultrasound deep down in the brain? Why should I think that's going to affect the brain in any kind of useful way? Turns out it does, and I'll kind of take you through the history of this a little bit because I think it's insightful to see how this technology has grown. The first person to notice this was in 1929. They noticed that if you turn ultrasound on to a heart cell, which the heart cells are kind of like neurons in some ways, they're excitable in similar ways, he could cause a frog's heart cell to react in different ways. So the voltages that he was recording change depending on if the ultrasound was on or off. That was the first time someone noticed it. Almost 30 years later, a couple of brothers who are famous in transcranial ultrasound delivered ultrasound to the visual system of a cat. What this is showing here is you can put electrodes on the back of an animal's head or a human head, and if you flash a light at their eyes, you can actually see a voltage signal as the brain responds to that flashed light. So what these guys did, the Fry brothers, is they noticed that voltage signal was suppressed if you were delivering ultrasound to the visual system at the same time as you flashed the light. And this has now been reproduced quite a few times in the modern era, but this was in 1957. They showed this in a cat brain. The reason it didn't take off at that time is shown by this picture. So this is their human device that these same guys came up with. They have 4 elements. Okay, so 4 elements of an array is not enough to get much of a focus yet. Your superposition effects aren't really kicking in a large way yet. You don't get a lot of gain. So it's not a great focus. And you need a whole room to control, right? This is the controller of this whole wall back here, just to drive 4 channels, right? So just didn't have the technology necessary to take advantage of this. Compare that to today. This is the Insightec Exablade device. It's 1,024 elements in this little helmet here. So factor 250 more elements. The whole thing's MRI compatible. You can put it in an MR scanner so you can actually look at someone's brain when you're doing stuff. And you get really nice tight focus with this. So now the technology has kind of caught up to this idea to where we can actually do this in a clinically relevant way. So because of that, in about 2010, interest in ultrasound neuromodulation started to spike again. For those of you not familiar with neuroscience, this is a raster plot. So this is counting action potentials of single neurons. So when the neurons communicate, they fire a voltage signal, and this is counting these action potentials that happen over time for a few dozen cells. And you see when you turn the ultrasound on, at the location of the electrode, you get this sudden surge in spiking activity. So where it's kind of random before, you get this surge, synchronized spiking, and then it goes back to normal. So this is showing kind of, again, coming back to something like what we saw with the Fry brothers, but just showing that we can change the way neurons are communicating with each other by delivering ultrasound. And so based on this, people started doing these experiments in rodents where you deliver ultrasound to the motor cortex. If you watch this little light, turns on when the ultrasound turns on, you can see the rodent moves. Right? So you're able to actually cause a behavioral change based on delivering this ultrasound. And so there's a whole bunch of studies that happened in the early 2010s based on this, looking at what's the right what are the right parameters to use, etc. How can we maximize our effect on the brain using this ultrasound? And the first in-human experiment happens in 2014. This Ben Lagon over at, I think he was at Virginia Tech at the time. He's at the University of Virginia. But he showed that if you deliver ultrasound to the somatosensory cortex, the part of the brain that processes sensory input, you can affect people's experience, like ability to tell the difference between whether you're poking them with like 1 pin or 2 pin, kind of subtle things like that, but you could have a significant effect on their ability to sense those things. So the field started to gain a lot of momentum. People are getting really excited, and then there's a bit of a crisis. So in 2018, there's a paper that comes out. They did this clever thing where and people do this all the time now, they put an optical window on a rodent, so you could use fluorescence to measure how different cells are reacting across the brain in real time. And so this is kind of the readout they get, and they're marking, here's the visual cortex, here's where you expect to see visual information, here's somatosensory cortex, this is what we were just talking about, where sensory input's coming in, and here's auditory cortex, this is what's processing sound. And what they showed was, there's basically, if you've got a lightly anesthetized mouse, and you deliver ultrasound to the motor cortex, or you play a sound, you buzz at the mouse, you get pretty much the exact same activation pattern in the brain. And the mouse twitches. So it's lightly anesthetized, it's awake enough to startle if you play a loud sound in its ear. So this raises a huge question. Are we actually modulating the brain, right? Or is the mouse just hearing the ultrasound somehow? Okay, ultrasound, usually in these experiments around 1 megahertz, that's outside of the frequency hearing of the mouse, but you pulse the ultrasound. It has a pulse repetition frequency. You're turning it on and off at some frequency, and that frequency is right within, it's usually on the order of a few hundred hertz or a few kilohertz. That's squarely within what you would expect the rodent to be able to hear. So this raises a big question, like, are we actually doing what we think we're doing, or are we just has this been an artifact all this science has been an artifact? There were a couple problems with this paper, most notably the particular regime they picked for their pulse repetition frequency. It was like designed to be audible. It was worse than a lot of the other ones, but it still raised a lot of concerns. So there were kind of 3 responses to this that I want to take you through, partly just because I think it's interesting to see the history, but also hopefully to convince you that we really are affecting the brain with ultrasound in the way that we say we are. So the first response was from a lab at Stanford, and what they did is they wanted to directly address this in this rodent model that people had been using. So they deafened some mice and had some mice with hearing, and they deafened them in all kinds of different ways. They actually brought in like an ENT to think of all the different ways the mouse could hear, all the different pathways that the ultrasound could make it to the ear, and make sure that all the different kinds of deafening that you would need to do to eliminate that pathway were done. And long story short, they were still able to produce this twitching behavior with targeting the motor cortex. And they also showed that you can eliminate the auditory response, which they could also reproduce by using a cosine envelope. So instead of just turning on and off in a hard way, you turn it on and off with an envelope, and then you eliminate the auditory effect. The second response was to look in a larger animal model and start seeing if you could get target-specific effects that lasted longer than just the ultrasound itself. So the first group to do this was a group in Scandinavia, and they did, I won't go into all the details of this just because of lack of time, but what this big plot is showing is that if I target 2 different regions, here and here, in an anesthetized non-human primate, so these are macaque monkeys, then I look a couple hours later, and the connectivity of their brains, the resting state connectivity measured with fMRI, has changed, and it's changed in a target-dependent way. So it's not just whether or not I turn the ultrasound on, it's where did I target the ultrasound? Did I target it here or here? And I get a different result either way. And again, this is hours after the stimulus, so the idea that there would still be a lingering auditory artifact is very, very unlikely. And finally, the last one I'll talk about is actually some of my own work as a postdoctoral fellow, looking at can we modulate awake behavior. So all these other studies are done in anesthetized animals. can we modulate behavior in an awake animal, also in a target-specific way? So to do that, in my postdoc here, I designed this, this system that's basically, this is a 256-element ultrasound array that can be attached to a macaque's head while they're playing a video game. So they're playing this little video game, they're looking at dots on the screen, and I can deliver ultrasound to their visual system while they're playing this game and see if I can change their behavior on the game in a target-specific way. And because it's an array, I can do electronic steering, so I don't have to move the thing to change where I'm steering. So for example, I can target the left nitrogenated nucleus. This is a visual system relay. So all visual processing system has to go through one or the other LGN, and I can target both of them. And one thing that's cool about the visual system, if you're not too familiar with neuroscience, is processing the visual system is divided in the brain. Right? So everything that I see kind of on the right side of my visual field gets processed by the left side of the brain. So if I affect the left LGN, I should only be affecting what the monkey is seeing to its right. And if I affect the right LGN, I should only be affecting what it's seeing to its left. So then we train them to do this really simple task. I'll give you a chance to do it. It's very easy. You focus on this point. Once the computer detects it, they're looking at that point. 2 more targets appear. And the monkey's job is to pick the target that appeared first. Very, very easy. And there's just a random delay between the targets, and of course which one comes up first is random as well. And this gives us a nice way to quantify their behavior. So here on the x-axis we have the delay between the 2 targets. Negative means the right target came up first. Positive means the left target came up first. And we can say how often, given that delay, does the monkey pick the leftward target. So this is kind of what you'd expect. When the right target comes on 50 milliseconds before the left, most of the time he gets it right. This particular animal has a preference for the left target, picks it more often than not, pretty much always gets it right when the left target comes on first. Then we can measure the same thing after we deliver ultrasound. In this case, we deliver ultrasound at the right LGN, and we see we increase their bias for the left. So there's a noticeable increase for the leftward target. Now when the targets come at the same time, they're just always picking the left target. And we can really nicely quantify this. We can look at this theoretical delay where they're equally likely to choose either target. So this is a sort of a point of equal preference, the delay at which they would pick either both targets with 50% probability. And we can see at that same delay now what's the probability they pick the leftward target. So it went from 50% to 77% for this particular experiment. And if we look at this over time, then we can see how long the effect lasts. So here we deliver 30 seconds of ultrasound, in this case the left LGN, and we see this increasing bias for the rightward target that peaks 15 minutes later and then starts to come back to baseline. So not only are we having an effect, but the effect is lasting for several minutes, and we can reverse the effect. So if we target the other LGN, then the monkey's bias switches directions. Lasts for some amount of time, not quite as long, comes back to baseline a little more quickly. But this shows us that we're getting a target-specific effect. It's an awake behaving animal. We didn't move the transducer or anything. It's really hard to explain this as an auditory artifact. So this is another response to that concern. And at this point, with these studies and several others, there's quite a bit of activity in the field in response to this other study. Very well established now that the ultrasound can indeed affect neural activity. Any questions up to this point? You can feel free to stop me if you guys think you want to spend more time on something.
How close to the monkey's head do these sensors have to sit in order to be able to deliver this ultrasonic reading that's powerful enough to affect them?
So the ultrasound is delivered, I wouldn't call it a sensor because it's putting the energy out. So this is the ultrasound array right here. These are much smaller monkeys, but so it's sitting like this. And the readout here is what their eyes are doing. We're doing eye tracking. That's the readout. Does that make sense? Okay, so because of this, now there's a bunch of emerging human applications. I'll just kind of quickly go through just to give you a sense of some of what's happening in humans. First study, this is someone with essential tremor. So that big helmet that I showed you earlier is actually, the purpose of that is not neuromodulation, it's ablation. So if I crank up the power a whole bunch of ultrasound, it delivers heat. Ultrasound carries both thermal and mechanical energy. And at that focal spot, I'll get a spike in temperature. So I can actually do surgery non-invasively with ultrasound in this way. And that's what that big helmet device is for. That's what that was designed for. So this lab took advantage of that existing device, and before someone went in for surgery to fix a central tremor, they turned the pressure way down, they turned the intensity way down, and without heating delivered some neuromodulation. And you can see this is like the magnitude of their tremor. That's what the blue and the orange bars are before and after sonications. So you see nothing really happens at first. Then they decided to move it just a couple millimeters onto what's called a white matter tract that was near the target that they were targeting to see if that would have a better response. And then it didn't have an immediate response, but 3 minutes after, the tremor was basically gone, and it ended up lasting about 45 minutes after they delivered that. So they're able to extinguish the tremor without damaging the tissue, and then they actually, this person was going in for the surgery, so they had to wait an hour because they need that feedback during the surgery. They had to wait till the tremor came back, which it did, and then they were able to proceed with the rest of the treatment. And this same study has been reproduced here at the University of Utah. I wasn't involved in it directly, but this is a study with humans at the University of Utah showing something similar. This is the raw data from the accelerometer on their hand, and you can see you turn the ultrasound on, it starts to come down until it is extinguished. And it lasts, depending on the patient, on the order of dozens of minutes. There's also a bunch of studies going on for pain. So this is actually in healthy volunteers. This is kind of a hard one to read. But these are two different targets, and then sham. And what they found is for the posterior insula, that's what PI stands for, it's a particular location in the brain. If you have a healthy volunteer, you put their hand on something that causes the sensation of pain, they report less pain during ultrasound targeting the posterior insula than compared to sham or targeting a different part of the insula. Maybe more interesting, another study here at the University of Utah using this diadem device, it's called. These are in patients with chronic pain, and this was it showing that with one treatment session, you're able to get a prolonged decrease in a patient's pain that's significantly greater than if you just do a sham treatment. Sham treatments in ultrasound are a little bit weak because there is this sound and there's some tactile experience when the actual ultrasound's on. So that's still an ongoing concern in the field, but these are great results. Similar, this is for depression, measuring change in depression score. Also University of Utah. Harvard's done a similar study. Again, this is sham. This is real ultrasound. They delivered multiple sessions here and saw a very significant decrease. Down is good here. I'm not going into the details of how they're measuring depression in this case, but they get a significant decrease in depression symptoms compared to sham after the treatment that persists for several weeks after the follow-up. And here they're also showing some changes in fMRI as well, which I won't go into detail on, but they're able to see changes based on the ultrasound, both in sort of more objective measures like fMRI and also the subjective measure where these patients are reporting their own symptoms on these clinically validated scales. Another emerging application is addiction. This is University of West Virginia. Patient comes in with high craving for all of these drugs. Deliver ultrasound of the nucleus accumbens, and this particular patient just extinguishes the craving. Lasted 90 days, and this patient did go into remission for addiction. This was not a sham-controlled study, so lots of these studies that I'm showing right now, they're really early days. A little bit this one's pretty anecdotal, still a very exciting outcome. And there's more epilepsy, showing a decrease in seizures, PTSD, anxiety. Pretty much, if you can think of a disease that is located in the brain and we kind of have a sense of what network is involved, someone's probably trying to treat it with ultrasound right now. It's a very hot field at the moment. Okay, so that leads to the question of why. Like what's happening? Why does the ultrasound how does the ultrasound interact with the brain? How do we explain this? So one possible answer is thermal. We said ultrasound carries thermal and mechanical energy. This is a picture from one of these ablation treatments. This is where the MR is detecting a temperature increase. So we can measure this. And we know that heat suppresses neural activity. If you've ever had a really bad fever, you kind of feel, you can feel a little confused. You can feel at least slow, right? So heat does suppress neural activity, that's known. So could it be that we're heating things up? This is unlikely, I'll talk about why in a little bit, but that's one possibility. Then there's the mechanical mechanisms. So this is an illustration of how an ultrasound wave propagates through a medium. The sound wave propagates through a medium. It's a compressional wave. So you get wave fronts that have high pressure. So if you kind of follow these, this is where the pressure is high. In between those is where the pressure is low. So that travels through. And if you focus on an individual molecule as it goes through, those don't move with the wave, they go back and forth, right? So you get this particle displacement that's oscillatory as the wave propagates through. So we have at least these two mechanical effects, this individual particle displacement and this pressure wave that's moving through. There's also something called the acoustic radiation force. So because there's loss in the tissue, as the ultrasound wave propagates through, it actually carries a little bit of momentum with it. And at the focus, you get a little bit of a displacement. It's like a little poke of a few microns, a few micrometers. So you're actually displacing some tissue. And that effect is happening at that, remember we talked about there's this PRF, we're turning the ultrasound on and off. That push is happening at that frequency. A couple hundred hertz, maybe a few kilohertz. That's a lot closer to the physiologically relevant time scale. So that's maybe a good candidate for causing our effect here. And finally, there's this thing called cavitation, where as the wave propagates through, if there's little microscopic bubbles, then you get the bubble will oscillate. And this can happen with any kind of bubble that can get drawn out of the tissue because of the negative pressure. Or it can also, some people have theorized it could be happening in cell membranes, which could maybe behave a little bit like this. So that's another possibility. So the first question to answer when it comes to mechanisms is, do we think it's primarily thermal, mechanical, or some mix of both? So some work to try to answer that. This is some work from a professor that used to be here at the University of Utah, is now at Washington University, in these really simple C. elegans animal models. Basically, as this thing moves, when it detects it goes there, it shoots an ultrasound pulse at it, and then it will reverse its direction. And what's nice about these is you can easily change their genetics, so you can they've got these simple neurons, and you can knock out the proteins that make them thermally sensitive, or the proteins that make them mechanically sensitive. So if we knock out the thermally sensitive proteins, we see no effect. They're just as responsive to the ultrasound as they were before, whereas if we knock out the ones that make them sensitive to mechanical effects, then they stop responding to the ultrasound entirely. So this, in a very simple animal model, this is some preliminary evidence that it's mechanical, not thermal. This was basically repeated in rodents. So there's these channels, these ion channels, which help the neurons fire these action potentials that are known to be sensitive to mechanical effects. They're called piezo channels. There's a couple of them. In this particular study, they knocked out what's called piezo 1. And we won't go into details here. The main takeaway is the blue is normal mice. The red is mice without piezo 1 in their neurons. And you can see the effect of the ultrasound is highly decreased in the knockout mice. It's not completely extinguished. There's still some effect, but it's highly decreased. So this is really strong evidence that these mechanosensitive ion channels in a mammalian brain are involved in the response to the ultrasound. So we've got pretty good evidence now that it's mechanical. Another thing to mention is we design our neuromodulation pulses to be really low intensity. We expect heating of almost always less than 1 degree Celsius, often even less than 1/10 of that. So any heating there is expected to be very minimal. We've got multiple models showing reliance on mechanosensitive ion channels. So we now have pretty much total consensus now that the effect of ultrasound on the brain is mechanical. Which of those effects we listed? Remember, there's pressure wave moving through, there's particle displacement, there's this deposition of momentum, there's cavitation. Remains an open question, but a lot of people are starting to come around to radiation force. I'm not totally convinced yet, and I don't think most people in the field are totally convinced yet, but that seems to be where the consensus is starting to come. Okay, so zooming out again, I want to kind of talk about where I think neuromodulation is and sort of the process of bringing a technology like this into real clinical use. I don't know if you guys have seen this before, but I've seen this described as a way that something that kind of happens to technologies as they come to being actually used. Something happens to spark interest, right? We see that ultrasound can affect neural activity. There's this huge spike of interest, everyone gets really excited. Something happens to make everyone think it's not going to work at all, and then we come out to some equilibrium, right? I think this is obviously it's a generalization, but I think it's a decent one. And I'd say ultrasound neuromodulation is about right here. Like I said, if you can think of a disease that exists